Muscles are the driving force of all movement in the body. The muscular system consists of three major muscle types. Cardiac muscles and smooth muscles function involuntarily, whereas skeletal muscles function voluntarily. But to understand those operations, we first need to get a grip on the anatomy of skeletal muscles, which involves fibers within fibers with a lots of layers. Basically, a skeletal muscle is constructed like a really sturdy piece of rope. Thousands of tiny parallel threads called myofibrils squish together to form muscle fiber. Those muscle fibers then form larger string-like bundles called fascicles, which combines to form a larger rope-like muscle organ like your bicep brachii. Overall, these bundles and bundles configuration make muscle tissue very sturdy. So that is the structure part of the story. If you want to get into the part about how it actually moves, well, there are rules. There are two main rules and they have to do with proteins. These rules are 1. Proteins like to change shapes when stuff binds to them. And second, changing shapes can allow proteins to bind or unbind with other stuffs. Now remember that tiny myofibril that bundles up to form muscle fiber? They are divided lengthwise into segments called sarcomeres, which contain two even tinier strands of protein, two different kinds of myofilament called actin and myosin. A sarcomere contains both thin filament made up of mostly of two light and twisty actin strands, and thick filaments composed of thicker, lumpy-looking myosin strands. Each sarcomere is separated by what's known as a Z-line at either end. A muscle contracting is all about sarcomeres contracting, bringing those Z-lines closer together. When your muscle cells are at rest, your actin and myosin strands don't touch, but they really want to. Specifically, that clubbed head myosin wants to get all up close in person with the actin. When this happened, which it will eventually, it's called the sliding filament model of muscle contraction. But at the meantime, the pair have some obstacles to overcome. Namely, actin is blocked by a couple of protein bodyguards called tropomyosin and troponin, which kept getting in the way. Luckily, these guys can be bought off with a little ATP and some calcium. Remember, ATP is kind of like molecular currency, contains chemical energy, and your muscles are all about converting chemical energy to motion, so they are always hungry for more ATP. Muscle cells also has its own version of endoplasmic reticulum, the cell's transport and storage system. But in this case, it's specialized, so it gets a special name, the sarcoplasmic reticulum. It is loaded with calcium pump which use ATP to save up a bunch of calcium ions. And it's also studied with calcium channels that are linked to voltage-sensitive proteins on the membrane of the muscle cells. Say, if I want to move my arm, my brain sends an action potential along the motor neuron into the synapse with the muscle cells in my arm. The receptor on that muscle cell are ligand-gated sodium channel. So when the motor neuron releases our old friend acetylcholine into the synapse, the channel open up and create a rush of sodium into the cell as a graded potential, which, if it's strong enough, causes nearby voltage-gated sodium channel to open. When the action potential travels down, it eventually triggers the voltage-sensitive proteins that are linked to those calcium channels in the cell's sarcoplasmic reticulum. When those channels are open, the calcium stored inside rushes into the rest of the cell. Remember the protein bodyguard troponin, which blocks the interaction of myosin and actin? It loves to bind with calcium. And remember, when stuff binds to proteins, the protein change shape. So the calcium binding on the troponin causes it to pull the other bodyguard protein, the tropomyosin, away from the site on the actin strand that the myosin really wants to get a pulse on. But the only myosin that can bind to those newly exposed sites are the ones that are ready for action. That is, the ones that have already grabbed a molecule of ATP and broken it down into ADP and a leftover phosphate. When the myosin has done that, it moves into an extended position, kind of like a stretched spring, still holding onto the ADP and phosphate and still storing the energy that was released when they were broken apart. So after all that, with the myosin primed for action and the bodyguards out of the way, the myosin finally binds to acting. When they bind, the myosin releases all that stored energy and the myosin changes its shape. It pulls on acting strength. In the process, it shrinks the whole sarcomere and contracts the muscle. With the energy spent, the ADP and phosphate unbind from the myosin. That unbinding caused a small change in its shape, which led a fresh ATP to bind there in its place. That binding caused another shape change, but this time it causes the myosin to release from the acting. Then the myosin breaks down a new ATP into ADP and phosphate, and the cycle starts over again. And meanwhile, those calcium pumps are working hard to restock the calcium in the sarcoplasmic reticulum. So they start grabbing the calcium that's floating around and causing calcium to unbind from troponin. When it unbinds, the resulting shape change puts the tropomyosin bodyguard back to place. You see, it's a circle. So that's the complete illustration of muscle contraction. Hope you all enjoyed it.